Alright, this is Science for the Ravens. Cellular systems. Let's start out with cells and microscopy. Biology is the study of living things, and if it's alive, it's built from cells. That's your starting point. Get this straight. All organisms are either eukaryotes or prokaryotes. Eukaryotes, like every animal and every plant that you can see, are built from complex eukaryotic cells. Prokaryotes, like bacteria, are smaller, simpler, and made of prokaryotic cells. Both types pack subcellular structures, which means specific parts with specific jobs. You will need to learn these structures. In an animal cell, you get the nucleus. It holds the DNA, the genetic material in chromosomes that runs the whole show. You've got the cytoplasm, which is a gel where most chemical reactions go down. And you've got the mitochondria. These are the power plants, the site of cellular respiration, loaded with the enzymes needed for those reactions. And you've got the cell membrane. It holds the cell together, controls what gets in and out, and its receptor molecules handle communication, like picking up signals from hormones. Now, what about the simpler prokaryotic cell? Well, forget a nucleus. Its chromosomal DNA is one long circular chromosome controlling everything, and it just floats free in the cytoplasm. Then it also has plasmids, small extra loops of DNA. These carry bonus genes, like those for drug resistance, and bacteria can swap them with each other like trading cards, you know. It's still got a cell membrane to control traffic, and it's backed up by a rigid cell wall. You can see these things with microscopes. A microscope uses lenses to magnify, making a small thing look big, of course. But more importantly, it increases resolution. That's the detail, the ability to tell two close points apart from one another. That's what resolution is. The light microscope, invented in the 1590s, lets you see the big guys, nuclei and chloroplasts. Nuclei, what's that? That's the plural of nucleus. The electron microscope, which was invented in when? The 1930s. That was a big game changer, and it shows you the fine details, like the inside of a mitochondrion. It's the only way to see the really tiny stuff, plasmids and viruses. What about a transition electron microscope, a TEM? That has insane magnification and resolution compared to a light microscope. But what are the disadvantages? Well, it's not portable. You can't carry it around. It's incredibly expensive, and preparing a sample is a complicated ordeal. So you can't look at anything alive with it. Finally, let's think practical. The number of structures in a cell depends on its job. A muscle cell burns energy constantly, so it only makes sense that it's going to be packed with mitochondria. A plant cell stuck in the dark has no use for chloroplasts. You adapt to the function or you don't survive. That's it. Let's now talk in more detail about light microscopy. The light microscope. It's the tool that lets you get in the face of a cell. Here's how to use one. Don't make any mistakes with this now. First, learn the parts of what you're using. You look through what? The eyepiece lens. You magnify with the objective lens. You'll usually have three, like a ×4, a ×10, and a ×40. Your slide sits on something called the stage, which is held down by the clip. And you carry the whole microscope by the handle. The lamp shines light up through your slide so you can see. Now, you'll have an image in view. You bring it into focus using the focusing knobs. They're your controls. Your specimen does not go on raw. It must let light through. If it's too thick, then you slice a thin section. That's step one. Take a clean slide, which you've cleaned. Use a pipette to place 1 drop of water or clear mountant in its center. This secures your sample. And then use tweezers to position your specimen on that drop. If it's transparent, you need to stain it, otherwise you can't see it. Iodine stains cytoplasm. Methylene blue, that's used to stain DNA. You need to stain it to see it. Now put on the cover slip. Place it at one end of your specimen. Hold it at an angle with a mounted needle and lower it down carefully. Press gently with the needle to squeeze out air bubbles. Trapped air will ruin your view. And now that you're viewing the specimen, clip your prepared slide onto the stage. So you're going to want to start with the lowest power objective lens. Remember, there's the ×4, ×10, ×40, so use the ×4 to begin. Use the coarse adjustment knob to raise the stage up to look to just below the lens, Then look through the eyepiece. Turn the knob to lower the stage slowly. This avoids smashing the lens through your slide. Stop when the specimen is nearly in focus. Then use the fine adjustment knob to get a nice sharp image. If you want more detail, switch to a higher powered lens. You're on the ×4, right? Okay, switch to the ×10 or to the ×40. But you will need to refocus. Once you do that. More power means a closer look, but a smaller field of view and less light. So work methodically. Okay, let's talk about the numbers involved with microscopes. Microscopy isn't just for looking. It's also for measuring. Magnification, I've said it earlier, ×4, ×10, ×40. That's how many times bigger the image is than reality. And you have two ways to find it. First, if you know your lenses, total magnification is equal to the eyepiece lens magnification multiplied by the objective lens magnification. Let me say that one more time. Total magnification is equal to the eyepiece lens magnification multiplied by the objective lens magnification. For example, let's say you have a times 10 eyepiece and a times 40 objective lens. Okay, this gives you a 10 times 40 or 400 total magnification. Simple. That's the first way to do it. The second way, if you don't know the lenses themselves, is you have to physically measure. The formula is pretty simple, pretty intuitive if you think about it. The magnification is the image size divided by the real size. The image size divided by the real size. But both sizes must be in the same units. Centimeters divided by centimeters, millimeters divided by millimeters, etc., etc. You have to have the same units. If they are not, convert them first. Okay, so let's do an example of that. Now, I said millimeters earlier. I hope you know what millimeters are. 1,000th of a meter. What about a micrometer? What's that? 1,000,000th of a meter. Okay, so let's say we have a real specimen which is 50 micrometers wide, and I'm looking at it under 100 times magnification. How big will the image be? The image size will be the magnification multiplied by the real size, 100 times 50 micrometers, which gives us what? What's 100 times 50? 5,000 micrometers. And now convert that to millimeters. What is 5,000 micrometers in millimeters? The answer is 5 millimeters. 5 millimeters. Okay, so I'm going to assume that you know about standard form, right? Like, how do I turn 0.00123 into standard form? 0.00123 into standard form. The answer is 1.23 times 10 to the minus 3. You need to just count the amount of zeros that you hear in the number. That's really all there is to it. Okay, so now I'm going to talk about DNA. DNA is the blueprint of an organism. It holds all the genetic instructions an organism needs to live and to grow, packaged into chromosomes. So, how is it built? DNA is a double helix, a twisted pair of ladders, Each strand is a polymer, What is a polymer? Well, a polymer is like a chain. It's like a chain. And the repeating subunits are called monomers. So a polymer is a string of monomers. In the case of DNA, the strands, the polymeric strands, the repeating subunits are called nucleotides. Each nucleotide has 3 parts: a sugar, a phosphate group, and a base. Let's hear that again. A sugar, a phosphate group, and a base. The sugar and phosphate group are the same basically every single time, but the base changes. And there are only 4 bases: A, C, T, and G. A, C, T and G. What do those mean? A for adenine, C for cytosine, T for thymine, G for guanine. Let's hear those again. Adenine, cytosine, thymine, guanine. Okay? The two strands are held together by these bases, and they link across the center. They link across the center, like rungs in a ladder, and they pair in only one way. A is always paired with T. A is always paired with T. Adenine is always paired with thymine. So C must always be paired with G. That's correct. Cytosine is always paired with guanine. This is called complementary base pairing. It's not a suggestion, it's the rule. This precise pairing is what keeps the structure together and allows the code to be copied. So I mentioned polymers and monomers earlier. What was a monomer? A small basic building block. In the case of DNA, it's like a single nucleotide. Remember, sugar, phosphate group, base. And a polymer is a large, complex molecule made by stringing many monomers together. Many monomers together make a polymer. DNA is a polymer of nucleotides. You chain them together, you get a strand. You twist two strands together, you get a double helix, and that's the shape of a DNA. Okay. Now let's talk about enzymes. Enzymes are biological catalysts. What does that mean? A catalyst is something that speeds up a reaction without being destroyed or changed or used up. So, if you do a reaction and you have a catalyst in the reaction, the catalyst will be the same before and after. It will stay the same, but it speeds up the reaction. Enzymes are proteins that speed up chemical reactions without being used up. They control the thousands of reactions inside a cell, like respiration and photosynthesis. This network of reactions is called the cell's metabolism. You could speed up reactions by raising the temperature. That's one way to do it. Why? Because things knock into each other more frequently if it's hotter. That's what will happen. But unfortunately, by heating things up, it will accelerate the good reactions and the bad reactions at the same time. You will also destroy the cell eventually. So, life uses enzymes. They lower the energy needed, allowing vital reactions to happen quickly at body temperature. Each reaction has its own dedicated enzyme. And each enzyme is a protein with a unique shape, coded by a specific gene. Enzymes are specific. Every reaction involves splitting or joining molecules. The molecules being changed is the molecule being changed is the substrate. The enzyme has an active site where it binds to the substrate, where it binds to its substrate. The substrate must fit the active site perfectly. If the shape doesn't match, then nothing happens. Okay? This is called the lock and key mechanism. The lock and key mechanism. The enzyme is not altered. It binds the substrate, facilitates the reaction, releases the product, and then it's ready to go again. Okay? This specificity is why they're useful in the first place. And we can use that in real life. Like, think about biological washing powder. That contains enzymes that catalyze the breakdown of stubborn stains like ketchup on clothing. And they work because the stain's molecules fit their active site. Okay? Okay, so I just spoke a little bit about enzymes and how they are, every reaction has its own like dedicated enzyme. So this makes me think that enzymes must be pretty specific and fussy, right? Indeed. They need everything just right to get involved in the reaction. They need perfect temperature, they need perfect pH levels, and they need just the right concentration to be effective. If you get it wrong, they will stop working. Okay, let's talk about how temperature is important for enzymes. Enzymes like things to be warm, but not too hot. There's a sweet spot, and it's called the optimum temperature. The optimum temperature, and it's where they work the fastest. A little heat gets things moving, molecules jostle about more, enzymes and substrates collide more often, and reactions speed up. But if you push it too far, then you will cook it, and basically the enzyme shakes itself apart and its shape changes permanently. That's called denaturation. Denaturation. The substrate won't fit the active site anymore. Game over. It won't fix itself when you cool it back down. Like, you've ruined it, basically. Every enzyme has its own optimum, and for human beings, the enzymes involved in our metabolic reactions, human enzymes, that happens to be 37 degrees, which is your body temperature. How convenient, right? Okay, that's temperature. What was the other factor? Oh yes, pH levels. What's pH? pH is how acidic something is. How acidic something is, or how alkaline something is. Remember, these are like chemical opposites. Acidic, alkaline, opposites, right? Okay. And the pH scale measures how acidic something is, right? Water is neutral, it's in the middle, its pH level is 7, okay, for water. Enzymes are really picky about the pH level that they operate at. If it's too acidic or too alkaline, then that messes with the bonds holding the enzyme together, and it changes the shape of the active site. It warps it, and that denatures it. It denatures it, often permanently. So, can you make any prediction? Well, most enzymes have an operating pH level of 7. Water, at the level of water. But what about pepsin, the enzyme in your stomach for digesting protein? that operates at pH 2, which is why you have stomach acid. Okay, so I mentioned temperature and pH level. What was the third one? Concentration. What does concentration mean? Well, it means having more enzymes in more enzymes in a unit of space. More enzymes in a unit of space. Okay, more enzyme molecules mean more chances for a substrate to bump into one and react. So, if you increase the enzyme concentration, you speed up the reaction, right? True, but not forever and ever, right? This only works if there is enough substrate to go around. If the substrate is limited, you'll hit a point where every substrate molecule is already being dealt with. And if you add more enzymes, they do nothing. So, I mean, think about washing up powder, biological washing up powder. If you just keep on adding more of it, your clothes, you don't get cleaner clothes like more rapidly. Like there's a limit. And the last one, that was enzyme concentration, and the other one is substrate concentration. This is basically the same logic, but flipped around. More substrate means a faster reaction, but only up to a point. Eventually, you flood the system, and every enzyme active site is constantly occupied, working flat out. That's the maximum rate. You add any more substrate past this saturation point, and it's useless. The enzymes can't work any faster. Okay, so what were those factors again? Temperature, pH level, and concentration for the enzymes. Now I want to talk about how you would investigate enzyme activity. So, here's how you measure the effect of temperature or pH or concentration on the reaction rate. The principle is really, really simple, and it's like at the heart of science, okay? Just change one thing at a time. Just one thing at a time. Change one variable, measure the rate, and control everything else. So keep everything else the same. Change one variable, measure the rate, control every other variable. Keep everything else the same. You can measure the rate in one of two ways. Either by timing how fast a product appears, or how fast a substrate disappears. Those are two possible ways to measure the rate of reaction. And the method adapts for any variable. So, for example, to test pH, you add a buffer solution at different pH levels to your enzyme substrate mix. Okay. To test substrate concentration, you vary the amount of substrate at the start. And then to test enzyme concentration, you will obviously vary the amount of enzyme. Okay. So I wanted to talk about catalase breaking down hydrogen peroxide into water and oxygen. I'll just talk about it very quickly, all right? So it's catalase breaking down hydrogen peroxide into water and oxygen. That's what I'm going to talk about. So you use a pipette to put a set volume of hydrogen peroxide into a boiling tube. Then you place the tube in a water bath at 10 degrees Celsius. You set up your gas collection apparatus. You add your catalase source, which is 1 cubic centimeter of potato, to the peroxide, and then you attach the bung fast. Then, this is a really visual experiment, so I can't really approve of me doing this. So I'm going to actually abandon describing that, because otherwise it'll just be lost. Let's just move on and talk about respiration, okay? Okay. So what is respiration? Well, in biology, respiration is how you get energy. And very importantly, it is not breathing. Respiration is not breathing. Breathing just gets the oxygen in. Respiration is the universal chemical process happening in every cell all the time that transfers energy from breaking down glucose. That energy cannot be used directly, so what happens instead is it makes ATP, adenosine triphosphate, ATP. ATP is basically the stored currency for energy, and it powers every process in the cell, but it's very complex how it does that, and we don't have time to talk about that here. But the whole operation, as stated before, is run by enzymes, which means its rate depends on the temperature levels and the pH levels. Respiration is exothermic. It releases heat. Your cells mainly use glucose, but they can break down other molecules too. Carbohydrates, proteins, or lipids. You use what you have. And there are two types of respiration: aerobic and anaerobic. Aerobic and anaerobic. Aerobic respiration literally means using oxygen. Aerobic respiration needs oxygen, plenty of it. For every single glucose molecule that's broken down, you get 32 molecules of ATP when you are engaging in aerobic respiration. So what is the chemical equation for that then? Well, it would be glucose plus oxygen yields carbon dioxide plus water. Right? That makes sense, doesn't it? Glucose plus oxygen yielding two waste products, carbon dioxide and water. So, wait. Do I know the chemical formula for glucose? Oh yeah, C6H12O6. C6H12O6. Okay, let me write that down. C6H12O6. What is it for oxygen? Well, I know the oxygen in the air. On a gas canister, it often says O2, right? That's right, because oxygen in the air is diatomic. Water, everybody knows water. H2O. What about carbon dioxide? The answer is CO2. CO2. Okay, so you're writing this down, and what do you have on your page? C6H12O6 plus O2 yields CO2 plus H2O. But this is unbalanced. This is very unbalanced. Indeed it is. So a beginner exercise is to try to balance that equation for aerobic respiration. And that's something I encourage you to do. What about anaerobic respiration? Anaerobic means without oxygen. It literally means without air, but in this context it means without oxygen. It's the backup system when oxygen is scarce. When you don't have a lot of oxygen, it will resort to anaerobic respiration. But it's inefficient. You only get 2 molecules of ATP for each glucose molecule broken down. The specific process will differ as well depending on the organism. Whether it's animals, plants and fungi, all have their own way of doing it. And you'll get those details in a while. Okay, now I'd like to talk a little bit more about respiration. Just a little bit more. Anaerobic respiration. This is what happens for you when you push past your limits. Let's say you are sprinting and your muscles are screaming and your body can't deliver oxygen fast enough to them, no matter how hard you are breathing. That's when you switch to anaerobic. In animals like you and I, anaerobic respiration partially breaks down glucose and produces lactic acid. So the equation is simple. Glucose is the single reactant and lactic acid is the single product. This acid builds up in your muscles and it hurts and it causes fatigue. The trade-off though is that you keep moving, but there's a debt to pay. When you stop, you're left with an oxygen debt. You keep gasping for air because you need that extra oxygen to break down the accumulated lactic acid and to restart proper aerobic respiration. That's the price of pushing anaerobically. Now, what about plants and fungi? Well, I can say for plants and some fungi, like yeast, they handle the oxygen shortage differently. In waterlogged soil, where roots can't breathe, or in a fermentation vat, they go anaerobic as well. And the equation is glucose as the single reactant, yielding ethanol plus carbon dioxide as the two products. No lactic acid for them. So remember, aerobic needs oxygen present, and anaerobic kicks in when oxygen is scarce, during a maximum effort, or in a waterlogged field. The substrate for both is glucose or another organic molecule you've broken down. And the products are different. During aerobic, it will give you carbon dioxide and water. And during anaerobic, it will give animals lactic acid, and plants and fungi give ethanol and carbon dioxide. But the energy payoff is everything. Aerobic is efficient. It nets you 32 molecules of ATP per glucose. but anaerobic being a last resort nets you just a measly 2. Just 2. You do it to survive, not to thrive. Okay, now let's talk a little bit about biological molecules. Biological molecules are the complex structures in every living thing. Carbohydrates, proteins, and lipids. And they are built from smaller basic units. Remember this. These molecules are your fuel. Carbohydrates, proteins, and lipids are broken down to transfer energy through respiration, making ATP. The energy your cells actually uses. And you need to know how they're built. First, carbohydrates. They contain carbon, hydrogen, and oxygen, as the name suggests. Their basic units, the monomers, are simple sugars, like glucose. You chain these sugars together into polymers and you get the large complex carbohydrates, starch, and glycogen. You can break these polymers back down into sugars by snapping the chemical bonds between the monomers. In your body, enzymes in the mouth and small intestine handle this digestion. You build them up, you break them down. Second, proteins. These are polymers as well, made from long chains of amino acids. And every amino acid contains carbon, nitrogen, hydrogen, and oxygen. Enzymes in the stomach and small intestine break proteins down. You build the chain, you break down the chain. Third, we have lipids, which is fats and oils. These are different. They are made from one glycerol molecule attached to 3 fatty acids. They are not polymers. They don't form a long repeating chain. Their elements are carbon, hydrogen, and oxygen. Enzymes in the small intestine break them down. And a key fact is that when lipids break down, the released fatty acids make the surrounding solution more acidic. They lower the pH. Okay. Let's now talk about photosynthesis. I mentioned respiration earlier, and now photosynthesis. If you don't know the equation, you don't know it. But here it is. Carbon dioxide plus water, using light and chlorophyll, makes glucose and oxygen. Let's hear that again. Carbon dioxide and water, using light and chlorophyll, makes glucose and oxygen. Instead of balancing this one out, I'm just going to say it aloud. 6 times CO2 plus 6 times H2O yields C6H12O6 plus 6 times O2. This is how plants and algae make their own food. They use energy from the sun or a lamp to build glucose. That glucose becomes their biomass, which is their living mass. And that biomass, that stored energy, is the foundation of nearly every food. Okay? Of nearly every food chain on Earth, rather, I meant to say. Animals eat the plants, other animals eat these animals, and photosynthetic organisms support almost all life. That's what I mean when I say it's the basis of every food chain on Earth. The process, this photosynthesis process, happens inside chloroplasts. Those contain chlorophyll, which is the green stuff that captures light energy, and the reaction is endothermic, meaning that it takes energy in from the environment, and it happens in two key stages. First, light energy splits water into oxygen gas and hydrogen ions. Second, carbon dioxide combines with those hydrogen ions to form glucose. Now, what about the rate? How fast this actually happens, and what does it depend upon? Well, surely it depends upon how intense the light is, right? For sure. Also, how much carbon dioxide there is, the concentration of carbon dioxide. And lastly, the temperature. The temperature. If you grow your own personal plant, you know this, that these factors matter, each one of them. Whichever one is in shortest supply becomes the limiting factor. It's the bottleneck slowing everything down. You measure the rate by the oxygen produced. More oxygen per minute means faster photosynthesis. Here's how you investigate it. Set up something involving pondweed. The oxygen it releases collects as a gas bubble in a capillary tube. Think of a very thin straw. Set up your lamp, your water bath, and your tube. Leave the weed to photosynthesize for a fixed time. The oxygen bubble grows in the tube. You then use a syringe to draw that bubble next to a ruler and measure its length. That length is proportional to the volume of oxygen produced. To test a factor like temperature, you repeat the experiment at different temperatures. You change nothing else. The light intensity, the carbon dioxide level, the time, all of that stays identical. The only change the one thing that you're actually testing. Only change the variable that you're testing. That's how you find the limit. Last, let's speak about the rate of photosynthesis. Photosynthesis does have a speed limit, and three things control it: light, carbon dioxide, and temperature. One of them will always be the bottleneck, the limiting factor, and your job is to understand each one. First, light. I mentioned light first. Light provides the energy. More light means you'll have a faster rate of photosynthesis, right? But this only holds up to a point. After that, the line on the graph flattens. Light is no longer the limit. Something else is. In an experiment, you change light intensity by moving a lamp. But listen, the relationship isn't simple. Light intensity obeys the inverse square law. This means intensity is proportional to 1 over the distance squared. If you halve the distance to the lamp, the light intensity becomes 4 times greater. If you triple the distance, it becomes 9 times weaker. You don't just plot distance, you either measure the light directly with a meter, or you calculate the intensity using this law. Second, carbon dioxide. CO2 is a raw material, the same pattern. Add more CO2, the rate goes up until it hits a ceiling and the graph plateaus. At that point, CO2 is no longer the limiting factor. If light and CO2 are both plentiful, then the limit must be temperature. To vary CO2 in a lab, you dissolve different amounts of sodium hydrogen carbonate in the water. It releases CO2. And the last thing I mentioned was temperature. This is about the enzymes. If it's too cold, the enzymes work slowly, and that's the limit. But if it gets too hot, around 45 degrees Celsius, the enzymes denature, the rate crashes. That's outdoors on a scorching day or in a poorly managed greenhouse. To control it in an experiment, you can use a water bath. So remember this. Don't blame it on the sunshine. Blame it on the CO2. Okay.